Do you mind if I put you two together to solve this problem? So do you guys have an idea?
For the not for the after.
Okay. יש לכם, פתרתם את זה?
אנחנו מנסים להבין למה זה ירצה לעשות דבר.
אוקיי, תכף נסביר, אבל... הבנתם מה היא עושה?
כן, פשוט זה יישאר דולק.
יישאר דולק, מה זאת אומרת?
זאת אומרת שמספיק לו אחד
כן,
מהארץ
כן,
בשביל לפעול.
כן. אוקיי. Would anybody like to... so what's the answer? I want to ask if anybody would like to say what the answer is. Yeah. So the answer is, your name, Omri says, it switches the asymmetry. This is a delay after the off step and no delay after the on step. Do you agree? Okay, so would you be willing just to draw that out for us? Okay, so let's give him some applause. Okay, so your name again? Omri, where? What faculty?
Physics.
And what stage?
Starting.
Starting PhD?
Yes.
Okay. So on step, okay, now we're, yeah.
Okay. On step, X start with same time scale.
Yeah. Okay. Let's even say that X star is really instantaneous, right?
Okay.
Okay. Yeah. Then Y?
Y starts.
Yeah.
And that starts as well because it's OR with X. So
That's right. So because of the OR gate, it's enough that one of them binds, right? So Z starts. Now let's do the off step. Then can you just do it like up here?
okay.
Yeah. Yeah. Off step. X goes off immediately. Y starts being diluted away.
Yeah.
Yeah.
And then
Z.
if this is K,
Yeah.
it dilutes afterwards.
Okay, so this is here is KYZ, right?
Okay.
KYZ. So I'm going to write it big here. So let's give Omri a big round of applause. Thank you very much. So there is a delay after the off step, and it's kind of nice for me to think about this as the OR gate. That Y is like an uninterrupted power source on your computer. So suppose you have your laptop, it's connected to the electricity, and now you take it out of the electricity. If you didn't have the battery, it would go right off, right? But since you have the battery, it stays on for a while. So Y is like the battery when you take off. If you have no battery, but you plug your laptop in, it goes on immediately. So this delay after off is an uninterrupted power source. When do you need such a thing? So E. coli uses this circuit many times, but for example, one time is when it needs to assemble a big machine, like for example, the flagellar motor. So when E. coli is in a bad situation, when the going gets tough, the tough get going, it starts building, making about 30 kinds of proteins that self-assemble into an electrical motor that rotates 100 hertz and pushes a flagella, which is a propeller. It's about 10 times bigger than the cell and makes it move at 30 microns per second. It's just 1 micron big, so it's 30 times its own body length per second in a fluid that's so viscous that the best analogy is like a corkscrew through cork. The Reynolds number is like that. Okay, so that's amazing. And the circuit that guides the production of the parts that make up the flagellar motor, which is made in steps, ring, rod, shaft, export apparatus, flagella, which is like a cylinder, and the subunits go from the inside and get assembled at the end. It's like amazing. Biomaterial, right? That's unbelievable. So the cell doesn't always make it, because making it is 10% of the cell's protein. The cells, if it makes it at the wrong time, will go 10% slower. It will lose the race. So only when you need to make it, those proteins, it makes those proteins, right? It's a lot of resources to be able to make this. It doesn't always make it. E. coli. And so it has this network where with signals like I'm starving, it's too hot, it's too cold, it's too much pH, too low pH, and start making the motor. Okay, that's the signals here. And then there, in fact, FLHDC has 6 different transcription factors that bind its promoter. It's one of those that it's sensitive to a lot of stresses. And then it activates FlyA, by the way, which is positively autoregulating. And with an OR gate, which is actually kind of like a sum. This is a sum. It activates the genes, for example, fly, L, M, N, open, operons that make up the proteins to make up the ring, rod, shaft, etc. And fly A then also, in later times, makes the subunit, makes the rotor. But let's be concerned with this feed-forward loop here. And experiment done by Shiraz Khalil, 2005, again, replace this here with green fluorescent protein. So there's just a copy of this with green fluorescent protein. So the cell can still make flagella, but it has a copy that's just reporting for this promoter with green fluorescent protein. And our control, we want to compare to simple regulation, is even a nicer control here. What she did is she went with molecular scissors and she cut out the fly A gene from the chromosome of E. coli. By, FLHD. So you have just simple regulation. So no fly A. Okay? This is the control. And when you look at how much flagella promoter is made as a function of time, you put E. coli, you turn on the stressful conditions, and for both circuits, simple and FFL, you get a turn on that's basically the same. And when you turn off, there's a 1 hour delay in the FFL. And when you think about this 1 hour, that's a time scale of making a motor. So when you think about it, if you've already invested in making motors for a while, and now E. coli swims already, and it goes into a good environment for a little while, you don't want to shut off. You give extra time to finish up that last motor. It takes like a couple of generations actually to finish a motor. So this provides E. coli with spare capacity to finish up the motors. So that's a situation where you use the OR gate. Any questions about that?
So you want to finish like in the middle of the motor?
No, you don't want to finish in the middle. We'll get to that later. It's actually, there's a turn-off order where the first thing made is actually the first thing turned off also in this circuit. But that's more advanced, so we'll get to that later. It's like a FIFO, first in, first out order. Yeah.
The switch between these two functions,
Yeah.
these two circuits,
Yeah.
it's a big change in the DNA of E. coli.
To go from this circuit to this circuit, you cut out one gene.
Yes,
Yeah.
but how does evolution does this?
How does evolution cut out a gene?
Yes.
So the question is, how can evolution cut out genes and make changes like this? So, in fact, one of the most common mutations, much more than changing a letter in the DNA, is to delete a region. That happens maybe 1 every million generations, as opposed to 1 every billion generations of changing a letter. So it's very easy to lose a gene. So actually, all the genes in E. coli are also kind of a miracle. They're retained for a purpose.
So the mutation of just losing the part or adding a part inside a section, it's
Adding parts and losing parts in biology, duplicating parts, is super common. So if you think of animals, the ability to even to duplicate segments and go from 2 legs to 3 legs to 4 legs in insects or something like that, based on these gene duplications. And when you look at our genomes, in many cases we have genomes that look quite a lot like yeast, except that if yeast has 1, we have 4 copies of the same protein that are specialized for different tissues. So these genome duplications occurred. So if you think the engineering here for making biology, you can use these circuits, modules, and duplicate them, modify them on the long time scale. I'm talking about hundreds of generations, right? An individual has 1 network. So that's very interesting. And understanding those dynamics is also very interesting. Look at the tree of life, how organisms evolved. There's this amazing synthesis called Evo-Devo, evolution and development, about how these networks, maybe we can play this song next time. Evo-Devo. Does anybody know this song here? It's a cool song. And, well, to understand how this logic guides development of embryos and animals. And things like that. All right, so if we think about the coherent type 1 feed-forward loop, the OR gate has the opposite sign-sensitive delay. And I want to tell you that all these 4 coherent FFLs have some kind of sign-sensitive delay. Either after on or after off, or it could be things that are a little bit different, but they all have it. Leaving open the question, why is the coherent type 1 feed-forward loop so special that it's much more often than the other ones? And I don't know the answer to that. I mean, I have some clues, but that's kind of an open question. All right, so that's the coherent feed-forward loop. And before we finish this class, we're going to do the incoherent feed-forward loop, which is a circuit of great power and can do many things. I'll touch like the basics here. It's really like a transistor, I think, of gene regulation networks. It's a wonderful circuit, and we keep discovering more things that it can do. And just like the coherent feed-forward loops, it occurs in E. coli very often, and also in all known gene regulation networks, in humans, mice, in the human kidney, the human brain, the human muscle, the intestine. Okay, so we're going to finish up with the incoherent feed-forward loop. This weird circuit where X activates Y and activates Z, but Y represses Z. So one arm activates and the other one inhibits. It activates an inhibitor. So actually it activates directly and inhibits at a delay. Yeah? So what does this do? So this is time. This is Z. So I put in my step of Sx. So Z starts being made. Y isn't there yet. So Z starts being made. And Y is... No, I apologize. I'm going to do it a little bit more slowly. So here is Y and here is Z. So Z starts being made, but when Y crosses its repression threshold, Z stops being made. So we get a pulse. In other words, the incoherent feed-forward loop, in a kind of simple way, is a pulse generator. It changes a step input signal into a pulse. It's kind of like a derivative. When you do more sophisticated mathematical analysis, it's kind of a logarithmic derivative, actually, with some smearing. That's just an aside that we'll get to later. All right, so it's a pulse generator. That's not it. That's not it, though. Maybe I should say right away. You see pulses in many biological processes where you, for instance, human cells, in order to grow, they need a hormone, let's say EGF, epidermal growth factor, and it goes into the cell, and it starts, in the end, make the cell divide, but that happens in a series of steps, and there's what's called early proteins that are made in a pulse, and they include, for instance, transcription factors for the next stage of proteins, etc. And so you get plenty of pulses like that in biology. And a lot of times you have an incoherent feed-forward loop there where transcription factor activates a gene and also an inhibitor of the same gene. In the EGF system, for example, that have been well characterized. So you have pulses very often in the cell. Yeah? Yeah, great. So the question is why they put here Z decaying to a zero steady state. Because I just wanted to start simply and to say this is what happens when there's strong repression by Y, right? So if Y is really like very strong repressor, it goes. But that's exactly where I'm going. What happens if Y is not such a strong repressor and there's a partial repression? Am I explaining myself? Okay. So when Y is a partial repressor, then we get the following situation. Sx, y, kyz, z starts being made. At this point, it stops being made, but it goes down to a lower level than it would if y was not there, but not all the way down. Because in this case, I'm assuming y is a partial repressor. So this is the partial repression. And what you can see, though, is something kind of cool. You get speed up of z. Speed up just like we saw in the negative autoregulation, except here there's no feedback. It's a feed-forward device. And what do I mean by that? I mean that speed up compared to simple regulation, right? X, Z, with the same steady state. So I'm making like a mathematically controlled comparison. I say I want to compare two circuits, an incoherent feed-forward loop and a simple regulation. So simple regulation with the same steady state looks like this. It reaches the same steady state. The reason that the feed-forward loop is faster is because, it's by definition, it's because Z starts going fast because Y is a repressor. If Y is a repressor, it has to go faster and then slower. And basically you get, it's like similar to the negative autoregulation. You get a fast motor, and then you have strong brakes because you activate the repressor. And then you can reach the desired steady state. Speed up. So the faster the promoter of Z is, the more you can speed up. Of course, yeah, okay, and this is in fact observed in the galactose system of E. coli. Similar experiments, you can compare incoherent feed-forward loop in the galactose system, and you delete the repressor, and it goes like simple regulation. Then you add the repressor, and there's this little overshoot, and you get a speed up to,
Can you explain again why it's faster simple regulation? Because before Y turns on, it's just there's only simple regulation.
Right, there's only simple regulation here, which goes to a high steady state. So here you get the benefit of this high beta, but a low steady state.
But the speed up doesn't the initial speed up.
Right, so you have the same alpha. So in the beginning you have beta over alpha. This would be this steady state. But you're getting to this steady state, which is beta prime over alpha, which is lower because of the repression. But in order to reach the steady state, you would need here to have beta prime for simple regulation. So it's a slower motor. You have to have a slower motor to reach the same steady state. Here you can have a fast motor and then repress it. Fast beta under. Yeah. So you can drive fast with good motor and strong brakes. So just to make a summary of the summary, we've seen so far in our course 3 ways to speed your promoter, right? The 3 ways to speed up. One is increase alpha, like degradation, which has the cost of making and breaking. Another one is negative autoregulation, which is great. You don't need to make and break, but I didn't mention it last time, it only works for transcription factors because they have to negatively regulate themselves. It doesn't work for the majority genes, which are not transcription factors, which are enzymes. So only for transcription factors. And then we have IFFL, incoherent feed-forward loop, which can work for any gene, gene Z, not only for transcription factors. And these can be used together. So you can take an incoherent feed-forward loop and add degradation, it goes even faster. Or put a negative autoregulation on the Y if you want to, and it'll go even faster. And that's, but I just want to make a comment, the network motifs can be nested inside each other. You can use two network motifs we understand already, and you can still understand this entire thing, because what the negative autoregulation would do is speed up here, and so you get faster. See, you can figure out, based on what I told you, these motifs made of two motifs. Did I explain myself? Yeah. And you can ask, I would say, my favorite example of incoherent feed-forward loops actually connect to what I started with when I keep saying about taking nice deep breaths, right? And talk about human stress and how stress affects our listening and our memory negatively. An incoherent feed-forward loop occurs in the human hormone system that controls stress. This is an amazing system we have. We share it basically with all animals going back 500 million years, where you have these glands in the brain, the pituitary, the adrenal gland. When there's a stress, the adrenal gland secretes a hormone called cortisol. Cortisol looks like a little cholesterol molecule. It goes into the cell membrane, doesn't need any pumps. And inside virtually every cell in our body is a transcription factor that cortisol binds to. It's called the glucocorticoid receptor, and goes to the DNA and activates thousands of genes when you're stressed. Okay, so this is like all over the body. It's just like in bacteria. You have a transcription factor, sugar binds here. It's cortisol binds, transcription factor goes to this. It's just in our cells, glucocorticoid receptor. And you know, this glucocorticoid receptor turns on genes, so when you're stressed, you see the lion, or maybe a friend told you something insulting. That's also causes psychological stress, yeah. Or you have low blood glucose, like it's before lunch now, maybe we're stressed. Or immune, like infection. What this does is, first of all, get you the energy to resolve the problem. So cortisol goes to the promoters and causes more glucose to be released to the blood. It causes amino acids to be metabolized into glucose. So you have a nice sugar, just the same sugar that bacteria like. We also like it, glucose. We run on glucose. Our brain runs on glucose. And we get really fast and good at making decisions and stuff like that. Blah, blah, blah. But that's what's called the acute stress response. That's what happens in the first hour. But if stress is continuing and you have chronic stress over days, weeks, you keep worrying about the exams, there's the chronic stress. And the same cortisol does other things. It turns on other genes. And now you have reduced memory, reduced learning. You stop making smart decisions. You basically run against the wall. So if something doesn't work, you just try it again and again, like that. It's the opposite, yeah? Chronic stress. You get a lot of high blood pressure, because high blood pressure is good when you want to escape from the lion, but it's not good for 30 years where you're worrying about your mortgage. And that's chronic stress. Okay, so inside the circuits that respond to cortisol in our bodies, there's these incoherent feed-forward loops and coherent feed-forward loops. And incoherent feed-forward loops are in charge of these acute stress, the lion kind of stress. They turn on the glucose. And then there's coherent feed-forward loops that only at a delay turn on other kinds of genes. So these incoherent and coherent feed-forward loops in this field of what's called hormone nuclear receptors is now realized to underlie a lot of the physiology that we know for these hormones, but just with a circuit that is very useful for differentiating between fast time responses and chronic responses. And of course, once you know that, you have some hints where you can intervene medically in order to not to lose the acute stress, which is the useful kind of, let's prepare for the exam, with the chronic stress. That we, since we basically have our psychology hooked up to an ancient stress system, it's not only lions, it's the idea that maybe there will be a stress. It's the prediction of stress that's causing us, right? And this is a lot of has to do with our psychology, how we interpret the world. And I think with this, let's take a nice deep sigh of relief. I'm guaranteed if you do that, you will lower your cortisol. And I very enjoyed this lecture, and I'll see you next time. Goodbye.